All right, and we are live. So we'll wait till people start to trickle in.
Yeah.
Jeff, take it away.
What's up, FCP fam? I'm a little sluggish today, so excuse
Me
my
too.
yeah. But yeah, we're just gonna have a chill show today. I think we're just gonna kind of shoot the shit with a handful of breeders and see where the conversation takes us. I will at some point bring up an interesting I don't know, was that a post or an article that you sent, Peter?
It was
Um,
just it was Susie B selections, I think, posted. Yeah, so Josh was actually fired up to talk about that topic.
oh sweet, sweet.
Um,
So I'll
just to
kind of
give people some context.
yeah.
He
So
posted not well. Actually, you know what? Let me just pull up the
yeah,
post.
I was gonna
Everyone
say.
can read along with me.
And
Uh,
I'll kind of give my commentary take on it, I think. And I may be wrong. That's why I'm kind of curious to see what Josh has to say about it. Because you really get perspective on this when you've taken stuff, past like F3, I think. When you start getting into like the F6 range, F7 range, you really can start to say, like, oh, yeah, this is pretty noticeable from here. But while Peter's pulling it up, basically, the from what I can just pull off the top of my head is when you're breeding with Afghan, specifically like the F1 generation, you're going to see primarily those Afghan germination and rooting traits, meaning like less water, drier climate, that kind of thing. You're going to see that behavior in the seed itself, and then in the seedling going into maturation. So, for whatever reason, from what I gathered, is when you make a cross with an Afghan, typically what you'll see first, especially if we're talking about like a hybrid cross, typically what you'll see first is the Afghan dominating everything as far as its genotypical traits, I guess, in the sense that it's going to be like a lot easier to handle drought type conditions. So
sorry. You know
let's
what? I think I can now.
blow it
Yes,
up.
transform. Yeah. Look at this. Uh, all
But
right.
it's
Well, you can. I don't know if you can. Can you read that?
yeah, my face will just be really close. It says, not sure if this is common knowledge or not, but if you're growing first generation hybrids of two very different varieties, take into account the mother, the female cultivar, when germinating seeds. Some Afghan based genetics, for example, germinate differently, have different requirements of moisture and humidity, most often less than their sativa relatives. This would likely be noticed when originally germinating the original P1 stock and won't change or have any noticeable difference until the second generation of said hybrid. For example, Afghan times Durban, first generation will germinate like Afghans. Afghan times Durban, second generation, germination will be variable. So for some
Based on
reason,
expression of chosen pairing.
yeah, so it says, for some reason, some people tend to think if an Afghan is pollinated with a sativa, the seeds created will immediately have attributes from said pollen. Is different seed shape and markings,
I.e.,
different
yeah,
germination.
i.e., different seed shape or markings, different germination rates, and so on.
But this, from what I understand, this stands with any F1 cross and they're calling it a P1 cross. From what I understand is that F1 generation is not as diverse. It leans more towards the mom inherently, and then when you go to the F2 generation, you see more phenotypical expression. So I was kind of taught like you can select from the F1, but you're going to get a way better idea of what's being passed on when you select from the F2. And I don't understand why, which is going to be really fun for me to learn, but I just understand that that's how it is. So it would make sense that if you're pollinating an Afghan mom, then the seeds are going to come from that parent, right? Like the seeds that come from that parent one, are going to resemble that mom. And then when you get into the F2 generation, which is two separate parents, and I appreciate that, because yeah, parent two would be the selection of the parents from the F2 generation. So you're not doing any back crossing into the first parents. Yeah, I understand why the P1 was used.
Nice.
Um,
Look at that.
yeah, that's what's up. So yeah, and that's one of the things like I respect about a lot of auto breeders is they're kind of forced to go to that. If you want any type of stability in your auto line, then you have to take it to at least F4, F5, F6. Otherwise, you get some weird expression in the autos.
I
Um,
think if I understood it correctly, it's basically the amount of water a arid desert Afghani seed needs versus or like you may just totally waterlog it versus a like an equatorial.
so that's what I was wondering is like when they're talking about like germination, are they talking about like less water and humidity? So basically you have a seed pretty much sitting on top of the soil, if not like barely nestled into it. And it has to
Or
be
in
like
it with just very minimal like
water.
spritzes of water rather than like versus like a
Right.
wet paper towel, right?
Yeah. Shit, that actually explains a little bit in what I'm doing. Because I'm doing like an Afghan. So the Afghan Hindu that I've got is F3 or F4 back cross once, and it's not nearly as vigorous as the Afghan times sour punch. And I don't know what the sour punch is, but like all the sour punches popped up first, and I was keeping the soil a little bit more moist. And all of the Hindu Afghan crosses took longer to break, and they're not like as vigorous. And I wonder if I need to back off the water even more.
Yeah, that was when I first read his post. I was thinking just the expressions of the first generation, but then I read it again and I was like, oh, he's talking specifically about germinating the seeds. Uh,
Yeah. Which yeah, I'm wondering. I've never like really experimented with like the moisture around the seed when germinating it. I know some seeds have like thicker and thinner shells, and they crack faster because the doesn't take as long for the moisture to penetrate.
yeah, the mother and the F1 is going to be the archetype seed personality for germination.
See, but like a full water, if you're talking Afghan, like a full water soak for 3 days, that's
I think he's
what.
saying you wouldn't do that to an Afghan.
Yeah,
You'd do
yeah,
that
yeah, yeah,
more to
yeah,
the
yeah,
like the Thai.
yeah. Which I mean makes sense, right? Like if we think about like we're using an Afghan that's evolved in Afghanistan for thousands or lived and adapted in Afghanistan for thousands of years, like it would pretty much be like put seed on soil because I Afghanistan is really close to the same like altitude and longitudinal latitudinal line.
As like
So
Humboldt or in U too, right in Colorado, right?
yeah, where we're at is me and my buddy were actually looking it up the other day. Like their light cycle is really similar. Their light or their temperature fluctuations are almost identical. Like they hit the same temperatures that we hit. They have like the same amount of rainfall that we get. So I would
Yeah,
assume that they're
Afghanistan's between 29 and 38.
yeah, and look up Grand Junction, Colorado. It's really close.
I'm trying to find a world map with like the line.
Oh, remember, dude came on with that overlay he had.
That was yeah, and I totally am blanking on the website that
Yep. But
uh
it in that, if their soil type is anything like ours, it's very compacted and very clay-esque. So in nature, those seeds aren't getting buried. They'll get like pressed into the soil, but they're not getting like buried. They would be very shallow. Um, and yeah, they would basically be taking any moisture that like evaporates up from any water table that may be nearby, or any type of groundwater that's coming through. Because it's wild how they search for them. There was some plants I had in my backyard that were on like, and these weren't even like indica dominant. These are like Afghan genetic. These were just like some random shit that I had. And the seeds germinated under the rock, like river rock, on top of weed barrier that's on top of like the hardest, most compact clay you've ever seen. And those roots ran underneath the weed barrier on top of the clay and stretched all the way to my grass, where there was like a tiny little bit of overspray from the sprinkler. So they were able, and that was like 10 to 12 feet away from where that overspray was. Not even to my grass, just to where that overspray that was hitting the rocks was. So yeah, those seeds popped up in like January. Just some random volunteers popped up January, and the roots stretched and hit the water that they somehow were able to sense was there, and thrive the whole time. Like they put all of their energy into those roots before the plant got like bigger than a couple inches tall. And then once it was getting like decent water, it just blasted off. So if the plant in general can source out water like that, I would imagine if you have genetics that adapted to a very arid climate like Afghanistan or Pakistan or Grand Junction, you would have genetics that are, I don't know how to say it any other way than designed to hunt out water, designed to react to any amount of moisture in the atmosphere or in the soil, like any humidity variation. And you can see it here, like where we're at. We're in the like getting into like monsoon type season. So it gets like more humid for a month or two here, and all the plants react to it. You can tell, like, as soon as humidity gets above like 15% here, all the plants are just like, bam. Even though it's 103 degrees here, all the plants are reacting to that humidity. So it's kind of wild. And my neighbor has like his whole front yard is nothing but like cactus and desert plants. And when the humidity picks up like this, they almost like swell. Like they almost all look thicker. And that's just from like excess humidity in the air. So I could see cannabis responding like in the same kind of context. Like, again, adapting and evolving to a very arid climate is going to give you very arid climate type traits, as reason would follow anyway. So yeah, that's my morning monologue. That's all I got. Yeah,
Sorry, everybody
elevation. I'm
is
reading the comments too.
fashionably late.
No, you're good. I'm reading some of the comments now. Like, yeah, elevation does make a difference in the plant. And I've been trying to figure out, like, it's an oxygen, it's like an air gas thing and a barometric pressure thing. Um, and it's wild when you like see those effects on a plant, like when you take all of the same indoor environment components. um oh yeah, dude, 100%. And I tell a lot of people this, like, and that's the thing, like if you look at the cultivation areas of Afghanistan and Grand Junction, there's like 200 feet difference. It's wild. We're so
Yeah.
close. But it's like when I send a cut down to my buddy down south, and he's running like all the same equipment as I am. So the only real difference is barometric pressure and the gases in the atmosphere. and maybe his watering frequency, but almost everything else is the same. Like you notice a significant expression difference, even if I match the temperature and humidity there. So like if we're running next to each other and he's telling me where his humidity and temperature is at, and I try and rock that same thing, the plant won't respond the same way his does. It's a gas. There's whatever gases in his atmosphere and whatever pressure from his atmosphere versus mine create a totally different response. And I don't know how that affects seeds. Right now, we're talking about like air pressure and what molecules are in that air reacting to how the seed, or like correlating to how the seed germinates. That's like that would be a trip where you basically like, dude, think about it. Uh, germinate seeds. We should do that experiment. Take one of those air pressure chambers where you can like change the positive and negative pressure and just
Yeah.
create a higher or lower barometric pressure and see what the germination effect is. Mm-hmm. Oh, 100%. It's like the one thing you can't get away from. Like you can't change the effects of elevation no matter where you're at. Unless the only other way is if you went to a specific place. This sounds so weird. You go to like Thailand and we're going to take just trash bags of air samples and we're going to run those through a lab to see like the ratio of gases in that area. And then you're going to note the barometric pressure through the entire year, and then you're going to build an entire pressure chamber with a gas input over your grow so that you can like inject the right ratio of air molecules and create the right pressure. Then I think you could get close to like maintaining exact replication no matter where you're at. But the cost of that it's stupid. But I think that's the only way around elevation. I really do. Like it's the one component that you can't control. We could try and replicate light in different regions. We could try and replicate temperature and humidity in different regions. We can even go and collect biology from different regions and try and implement that into our soil. But you can't fuck with the air. You can't. I mean, you can filter it and inject CO2, but you're not gonna you're not the pressure. It's like being underwater. The pressure you can't remove. So it's kind of a crazy concept that I don't know. I wonder in all the pheno hunting labs out there and all the people who are like sourcing out and selling these hella like pricey cuts. I wonder if they even like talk about where they hunted the cut at. Like we selected this at this elevation in these temperatures, like under a nutrient regimen or a living soil regimen. I've never bought clones from like a clone site, just from like buddies and stuff. So I don't expect that, but at what point does a $1,500 clone like toad venom come with instructions?
Right. Yeah, this was another one Dwight jumps on. Hold on, let me. Where are you? There we go. This I thought someone posted this yesterday.
Oh yeah, I've seen that a
Yeah,
ton.
the best strains from the 70s.
Yeah,
Look at that bag appeal.
I know. You know, I wonder about that too. At what point did we associate like visual with quality and then lose whatever those had? Like we often talk about, like when you breed for a specific thing, you lose something else. So if you're like really trying to breed for that bag appeal, like are you losing other compounds that shit had that none of us that started smoking in the early 2000s could possibly comprehend. You know what I mean? Like late 90s, early 2000s, like what was out in the 70s is not the same. Like on a chemical point.
Sorry, let
Sounds
me
good.
see.
Oh, you're good.
Give me 1 second. For some reason,
Like we often talk about, like when you breed for a specific thing, you lose something else. So if you're like really trying to breed for that bag appeal, like are you losing other compounds that shit had that none of us that started smoking in the early 2000s could possibly comprehend? You know what I
What
mean?
the
Like late 90s, early 2000s, like what was out in the 70s is not the same. Like on a chemical point. What the hell was that?
sorry, let
Sounds
me
good.
see.
Oh, you're
Oh,
good.
that's me. Hold on. That's
I was like,
us.
oh, oh bro. Oh, I thought I
You
was
could, yeah, you
fucking
could hear that,
tripping.
right?
Yeah, I thought I was tripping. I was like,
I didn't know.
there's
I
that
was
one.
like, I was like, who is that? And then I was like, wait, that's me.
I was like, I just said that. Oh, wait, that's me. Oh, shit.
Oh, look who it is to answer all our questions.
Oh, let's
There
go.
we go. All right, hold on. Let me. What's up, Josh?
What's up? What's up? How's everybody doing?
Good. How about you, man?
Good. We've been spinning our wheels because neither of us can actually answer these questions. Hold on. Do we see Josh's? Oh no, wait. Sorry. I'm trying to show Josh's little icon. Oh yeah, audio avatars. There we go. Boom.
Bambi.
There we go. So yeah, we were just while we were waiting for people to trickle in, I posted that screenshot I sent you about this. So what we're both understanding is he was even talking at the seed level that like the seeds from something that maybe comes from like the deserty kind of arid climate don't need as much water to germinate or should not have as much water to germinate as something that maybe grows in like monsoon type climate. And I think Josh muted himself. There he
Sorry.
is.
Yeah, I think there's definitely something to that. And since you shared that with me, I went in and was doing a little bit of research and actually found um, some, published white papers that kind of back this up. Um, and it appears to be that there are a set of genes, obviously, that regulate the metabolic processes that the seedling embryo goes through during the germination phase. And those, are obviously, as any other genes, inherited from the parents. And so I think what that statement that you had sent, I think it was like a Afghani Durban cross or something like that. Um, we're saying that it would be the parents that exhibit the differing characteristics of environmental needs during germination. Then that F1 generation um, would mostly be the same. But if you look at the Mendelian distribution of genes in an F1 population, provided that the parents are true parental strains, true breeding, you get a 25% distribution of um, offspring that are more similar to either the mother or the father, and then 50% that are a solid mix, right? A 1 to 2 to 1 ratio. The F2 generation, so a combination of two of the F1 offspring, would be really determined by the traits that were, I mean, if you did like an open pollination, that would be one thing. But if you selected two specific individuals from that population, the recombination of those genetics would resemble whatever those two individuals brought over with, from the parental lines. So that could be, if it's an open pollination of the F1s, the F2 generation are going to be a wide variety of trait types, with a much, that's why we like to pheno hunt the F2 generations. If you're a pheno hunter and you want to see the greatest number of traits that can result from any cross, that F2 generation is where majority of the plasticity and diversity exists in gene expression. And for many different reasons other than just the typical ratios and seeing things that, are latent genes or genes that pop up, recessive dominant genes, recessive traits, all that stuff usually shows up in the F2. But I'd never really spent much time thinking about, germination, the genes that impact and affect germination. And but here recently, I've had customers that are like, "Hey, I'm not getting good germination. I don't know what's going on." Then I, talk to other folks that are germinating the same strains, and then I've germinated the same strains. And I'm, seeing 85, 90, 100% germination rates. Others seeing 85, 90, 100% germination rates. That they're getting like, 20% or 10%.
Yeah.
And one of the things that I find is a lot of times when you get low germination rates, it's oversaturation. The seeds are essentially sitting in water and creating an anaerobic environment.
Yep.
Um, and
That's what
they
I
just
noticed. Yeah,
yeah,
that's what I noticed. Anytime I have like failed germination rates, it's typically too much water. And that's so I think we've all like kind of dialed in a standard that seems to give us successful rates. Cause I mean, I've gotten 20-year-old seeds to germinate with that. But then I'll notice like I'm doing some like some Afghan Hindu and some Afghan sour punch. And I know the Afghan Hindus into like F3, which made me wonder. I'm like, those took longer to pop. And I wonder if it's because my standard moisture was still too much, where it's very much like for what I normally do, that was too much water for the Afghan Hindu cross, you
yeah.
know?
I think most cannabis in general requires a lot less water than we typically think for
Yeah.
germination. And just in general, like, I mean, I've seen seedlings that are, 2 or 3 weeks old, um, where either for some reason or another they either got too hot or somebody forgot to water them or whatever. And I come in and I look at them like, well, crap, these are just gone. Like they're just dead. I mean, literally, those kind of like first set of fan leaves will be like brittle and crispy, and it's like, well, shit, maybe I've got something in there that I want to try to save or just to see. And you toss them a little water, and 2 or 3 days later, you're like, huh, how in the hell did those survive? Like they can't,
Yep.
they came back from a level of drought stress that now I don't know, that might trigger a whole set of genes that affects the plant the rest of its life, but it survived. Um, and so I think, overwatering, whether that's in vegetative stage, flowering, or during germination is a problem because this plant has evolutionarily developed to exist in some of the harshest environments on the planet.
Yeah. Oh, yeah. Well, and again, this is all anecdotal because I just kind of observe my surroundings in a way, I guess. But in I have this theory that in higher elevation climates, typically less water, and I may be wrong on this, but less water equals more oxygen to the root zone. And so when you're higher up in elevation, you need as much oxygen to the root zone as you can get. So you sacrifice a little bit of water uptake, especially if it's dry, up and hot up top, like you're not moving water as fast. You're trying to store it as much as you can. So there's this correlation between, water and oxygen at the root zone when we talk about like how how much a plant can take genetically, right? Because all of those things, like you're talking about, even from, starting a seed in a too moist environment, like how could that affect the plant permanently or impermanently down the road? I think there's something to that because the plant may be trying to adapt for this concept of suffocation. I don't know. My brain goes down these rabbit holes because I see certain things that correlate. Like for us, the higher up higher up in altitude we go, the less oxygen we get to our bloodstream. So that has to affect plants as well. And we know that because of
Yeah.
the tree line in the mountains, right? Like there's a point in the mountains where all vegetation just stops because there's no oxygen. So as we
Yeah.
increase or decrease oxygen at the root zone, what does that cause the plant to behave like?
Yeah.
And I
So interesting. What you're bringing up kind of ties into a pretty cool research experiment that I'm partnering with a company called Tavcana, and we're sending um, cannabis seeds into space for 9 months. They're going to be launched on a rocket. They'll be put into orbit. They'll orbit the Earth for 9 months, and then they're going to come
Space
back, and
weed.
we're going
Sorry, I
yeah,
got
we're
you.
going to space weed. We're going to eventually germinate those seeds. Hopefully, they'll germinate after being exposed to space radiation. But we want to see what potential impacts um, the space radiation might have, but also microgravity, as well as you know what genes are impacted. And it very well could be that genes that regulate germination could be impacted. And the first one is one that enforces dormancy, and that's the ABA pathway. The ABA pathway, which is the abscisic acid pathway. So before a seed ever encounters water, ABA levels are really high, and that's telling the germplasm, hey, there's no water here. There's the germination parameters aren't within the right range. We don't have what we need to germinate, and it triggers the seed to stay dormant. And there's a whole specific set of gene families that help to respond to dormancy. There's um, the CS pile gene, which functions as like intracellular ABA receptor, and there's a lot of studies that have been done from what I found just kind of looking through some of the research that focused on like hemp seed and hemp seed germination, that specific genes like the CSPYL5 um, interact with some downstream phosphates to block germination when conditions are either too dry or too wet. So it doesn't have to be that it's too dry or too wet. If the environment isn't optimal, that CSPYL5 gene complex will tell the seed, hold off, don't germinate. And then there is another complex. Um, Let me look at my notes here. What was it? It was the CSPP2C. It's a protein phosphatase 2C. And there's a couple others that um, act as negative or positive regulators on the ABA cascade. So what essentially what they do is they function like a biochemical circuit breaker. Is from reading about this and learning more about it, it seems like these genes, the ones that specifically like the CSPP2C and the protein phosphatase 2C, they act like a circuit breaker, and they shift activity depending on how hydrated an embryo becomes. Now, that doesn't mean that exposing it directly to water is going to trigger anything, because if it's too wet, it could be just as inhibiting as being too dry. And then you have another one, which is just as important, which is the ABA5 or CSABI5 gene, I've seen it expressed. I think I looked at about 6 different papers that had this gene that they were referencing, and not these pathways are also present in other plants as well, not just cannabis. So we can make some um, inferences about germination and the genes responsible because these exist in other dicot plants other than just cannabis. But there are some that are cannabis specific that we should call out. But
I saw somebody in the comments said they actually have sent like cherry tree seeds to space and such. So like
yeah,
they have,
yeah,
yeah, they have studied that in other plants.
yeah. So back in when I was in grad school, I worked with the Canadian Space Agency and NASA, and we did they had a big program back then. I don't even know if it's still going. It was called the CLES program. It's a
Was Neil
closed loop.
Iorio there when you were doing that? Does that name ring a
Really,
bell? He
man,
was a
there was so many.
NASA lighting scientist, and
I,
then he founded one of the lighting companies. But I remember interviewing him years and years ago, and he was talking about his research of, you know, getting plants to Mars, right?
yep,
Because you
yep.
got to eat on the way, and then once you're on a planet like Mars, kind of doing all that type of research. And I was like, this is so cool.
Yeah, Neil probably was doing that back when I was there, especially if he was involved in doing the LED lighting development, because a
Yeah.
lot of the LED technology that we are utilizing today came out of the space program because it was practically impossible to use incandescent lighting technology on the space station, or even like a module that got sent up. Like I know one of the first modules that we sent up were mung beans. And it was specifically, we used this aerogel, which was this like really light, super light substance. It was a matrix that they had originally created to capture space dust on these arrays that were fanned out to just float through space and capture various particles that were, floating around in empty space. But we were able to use that same gel because it was super lightweight, and weight's a really big factor when it comes to launching things into space because of how expensive it is, to get something up there. Like fuel costs for
Yeah.
a trip to space, it's pretty expensive. So we wanted to reduce weight as much as we could, but we put mung beans in a module. We used LED lighting, which was kind of crude at the time, but the diodes existed and it worked. You have more powerful LED diodes in a flashlight now than what we had then, but you don't really need a ton of light to get a plant to germinate. Really, you don't even need light at all. And what we were specifically looking at was gravitropism. We were curious at the times, like, well, if we send these seeds up and they germinate in microgravity or in a very low gravity environment, will the root and shoot know which way to go? Will the shoots know to go down, or shoots know to go up, and the roots know to go down? What we found was it in microgravity, sometimes yes and sometimes no, but light was a differentiating factor. If you expose the seedling to light, the shoots always went the way they were supposed to, and the roots went the other way. But if you did
So
not
so
expose them to
in the
light,
absence of gravity, light did the job.
light took over. Yeah, light was the signal the plant was able to use to say, oh, hey, this part of the plant goes this way and this part of the plant goes that way. Otherwise, without the presence of light and in microgravity, some of the seeds essentially turned into what tissue culture calluses. Really, the cells were, for the most part, undifferentiated. They would multiply and grow because they had all of the nutrients required for growth that were there, but they didn't know which way to go. And so the plant cells could not differentiate, hey, do we need to become a shoot or do we need to become root? And so it's pretty interesting stuff. And that stuff has come a long way since then. That was the early 2000s, like 2004, 2005, when we were doing that. But going back to cannabis germination, like there's two main pathways. There's the ABA pathway and then there's the GA signaling, gibberellic acid, or GA3 signaling pathway. So the ABA pathway promotes inhibition or keeps seeds dormant. The GA3 signaling pathway is what calls the seeds, once water penetrates the seed coat, once this inhibition occurs, the genetic balance violently flips towards GA production. And there's one trick that I've seen a couple of old timers use for germinating really old seeds that have a hard time popping, or maybe you have seeds that are old that do germinate, meaning that they're able to crack the seed shell and they'll stick out like the taproot, but then they just dud out right there. Usually
That's the saddest.
it's heartbreaking, because you see
Your
that
hopes go way up, and then they plummet.
and then it's all over, right? Like, it's just like the gibberellic acid at a low concentration usually is enough to trigger the needed enzymatic processes, to finish out that germination process.
I have read
it'll
that all over the place. Everything for old seeds is gibberellic acid. Like, that's the answer to recovering that.
So give us the, is it like X parts per however much distilled water or something, or?
Yeah, it's a very low, it's a low concentration of gibberellic acid. If you use too much, you can cause damage.
But are you mixing it with distilled water, or can you use just regular water, or what would you?
My personal recipe for old seed germination, it's kind of a multi-step process where I use H2O. I use first I'll do a sterilization wash to make sure there's no pathogens on the seed coat.
Of straight H2O2?
No, I use a diluted H2O2 and soak for 12 hours.
But do you do the 3% with how much water?
Well, the first step is a 2-to-1 hydrogen peroxide to water. Then I move that over to like a wash oxygenation, which is the 1-to-1 3% to distilled water. And then I move from that over into kind of a modified slurry. So instead of taking the seedling to a tissue culture media, which would be in like an agar gel, I'll use a very similar recipe as the seed rescue tissue culture media, but it's just in a liquid form, and I'll let the seeds soak in that, because usually at that point the soak in for 12 hours and then another 12 hours. So we got 24 hours of soak in H2O2, and that's multifaceted. That's not just a disinfecting stage, but it's also to create oxygen for the seed, because that's one of the problems that can happen with seeds that are stored for a long time, is they lose oxygen, which is needed for germination. But it does have that slurry that I have does have a small amount of gibberellic acid in it. And that is, I've found critical.
So that's where you're adding it at that stage.
Yeah. Okay. It comes after the hydrogen peroxide disinfectant wash, and then I used to use 10% bleach too. I would do a 10% bleach soak very briefly, and then move it over to just the 1-to-1 H2O to H2O mix.
So the bleach soak would be the first soak.
Yep, that's the first disinfectant.
And then the slurry, are you making your own, or are you buying?
No, I make my own. I mix up lab grade reagent chemicals with I'll put a specific amount of potassium in there, usually in the form of potassium hydroxide. I'll use phosphorus. I use a little bit of calcium, and so I get a very mild nutrient mix in there so that if the plant needs any nutrients to sustain enzymatic and protein growth, that is there as well. I put simple sugars in there. I usually have a little bit of fructose, a little bit of glucose, and then the gibberellic acid, and that's usually enough to give the seed everything that it needs to germinate. It's not a super complicated mix; like it's pretty simple. There's a little bit of
So,
nitrogen, a little bit of calcium, phosphorus, a little bit of magnesium. Um, I use I don't tell many people this, but I use like Thrive Alive V1, the thiamine multivitamin. I'll do to a cup of water. I'll do 1 drop of that. Um, that has a nice mix of micronutrients, plant hormones, and some vitamin B1, which can help cell elongation and plant growth. And then I think it's half a gram of C90,
mhm.
which is another mix of micronutrients
This
in there
is
as well. So
So,
this is
it's really what it,
quite a recipe.
right,
yeah. Well,
and
I
I
mean,
love it.
yeah.
well,
And so
unless
what I normally do is I'll make if I'm going to be germinating a lot of seeds that I know are old or need embryonic rescue type. Now, depending on the value of the seeds, some of those I just go straight to tissue culture with. Like I don't even fool with trying to do the rest. I just tissue culture program. The others, though, like if I have a lot of seeds, I'll make up um, base solutions of each of the inputs and then just mix those together at the specific ratios needed. Um, there was a company, Canamatrix, I think. No, not what Canamatrix. What was the name of the company? They made a soak for like a germination soak that was popular. I don't know if they're still making it or not. Canonomics, was it Canonomics? I
Yeah,
think.
as you listed off like the 18 things, I was like, I would love to just
well,
get it from someone else.
so okay, so
Yeah,
everything I'm listening to is basically like, from what I understand, it's like piecing together everything you would find in a natural seed solution. So, like, if you do a slurry of microgreens that you just popped up with like some legumes and other seeds that have
Some
natural gibberellic
aminos and
acid, the
enzymes and
aminos and
yep.
enzymes, and they've got some plant-based nutrients. If you create a solution of that and use that as like your third step, right? You can go through the entire hydrogen peroxide wash, and then your third step would be going into that
the
seed
energy
slurry
injection to
solution.
the heart.
Yeah, so for everybody at home who's like, that's way too much, in my mind, I'm like, get a bunch, like, start a bunch of seed sprouts and then follow kind of the K&F seed slope solution. Yeah, so you
It
end
was
up with
no
some
that
aloe and yeah,
that
yeah,
was the first. That was the first. What my current recipe, how it exists now, didn't start as like, oh, I'm going to go in and figure out exactly how many micromolars or millimolars of or milligrams, nanograms of
Of
all this shit
each.
that I need.
Yeah.
It started out with me taking about 200 hemp seeds that I had just laying around and germinating those, and then popping all the little seed coats off of them and blending those up and mixing them up and straining them and filtering them and soaking seeds in that
yep,
germination soak.
yep. And it doesn't
In
even
their
have to
own
be hemp
kind.
seeds. Yeah.
Yeah, because
Well,
my
and
thought was like, okay, if I've got seeds that
Yeah,
are
that's
germinating, that are healthy, that have all the stuff that they need from their own kind, then I know that what they need is there. And
yeah,
then eventually I figured out like, okay, what are all these parts? Because seeds at the time were, even hemp seeds were, $1 a piece, and so
expensive.
it's like,
Right.
it and
And that's what's
so
cool about
just
other plant study is you can figure out like, oh, these seeds are higher in this and these seeds are higher in this. So you can do the same concept, and the same way you're piecing together like certain chemicals individually, I think you can identify that in certain seed starts where it's like, oh, okay, I'm sunflowers contain a shit ton of oxygen You know, so if I do a bunch of sunflower starts and then blend those up, my water is going to have a higher amount of oxygen present in it, along with X amount of amino acids and whatever. So I think, you know, again, just as like on a home gardener kind of mindset, you can still do this
Yeah.
curation, I guess, with
It may
different
not be as
seeds. Like
perfect as
exactly
the more complex solution, but it's
right,
better than not doing anything.
right,
There's one
and
one step of the process I forgot to mention, which is the scarification process. So usually
yeah,
I'll take a little box, I put some 200 grit sandpaper in the bottom of
Yes.
that and just roll those seeds around kind of carefully. Um, and the point of that is cannabis does have a thick and pretty tough seed coat. So if over time, that seed coat, because of like loss of oxygen and oxidation, forms a polymer that's almost waterproof. And so, if you scarify that and create the little micro scars to where the water can soak through easier, it makes it a lot. You get a lot higher success rate.
If you're really concerned, are you just peeling that layer off altogether? Although I guess then
Yeah,
you're going into tissue culture, right?
yeah, I mean, if I've got something like really old that's really valuable or really rare, I'm probably cracking that seed and pulling out the germplasm and placing that directly into a media that's designed specifically for embryonic rescue, which is practically just a gel version of the um, hormonal activation process of that seed germination. So for me,
And
the
then
process looks like
just quickly, in that scenario, are you doing a quick hydrogen peroxide dunk,
Um, no,
or just
because
straight to
the
the
straight to the media, because the
okay
media that I mix up has specific bactericides and
anti?
fungicides. It's a it's a
Okay,
it's a sterile mix that inhibits growth of potential
so that's
pathogens or
solving that problem. Got it.
yeah. But on other seeds, I'm not tissue culturing. The first step is the scarification, then the chemical pre-soak for disinfection, then the hormonal activation soak, and then that last step is the oxygenation, paper, if you will. Kind of the old school matchbox trick, with the
Damn.
scarification. And then the next step is that softening disinfection pre-soak. That's really important because one of the most common pathogens that will destroy your seed germination. And usually we'll see it because the seed will start to germinate. You'll get that little tail poking out and then it stops. And then you pick that thing up and you notice that the tail just turns to mush. That's Pythium. And Pythium can stick around for a very long time. And it is a fungal pathogen that is prevalent. It's everywhere. It's in the air. It's on our shoes, on our hands, and so being able to disinfect against things like Pythium, cup of distilled water, even just a teaspoon of 3% hydrogen peroxide will work. And then I also like to add sometimes during that stage 2 or 3 drops of like a fulvic acid or a kelp extract. That will add beneficial trace minerals that can aid in germination, and so you're kind of
Yeah, I
preloading.
always use bioag.
Yeah, that's a great mix. So you just drop those scarified seeds directly into your solution. You put your glass somewhere or container in a dark, warm environment. I usually try to stick it in like a cabinet next to the refrigerator, but anywhere dark and warm will work. You let those sit for 12, 18 hours, but don't go more than 24. I don't go more than 24 because you risk literally drowning the seeds. And that's a big problem I see happen with a lot of people who try to germinate seeds like in a cup. They'll just toss like 10 seeds in a cup of water and they'll leave them in there for 3 or 4 days. They'll be like, oh man, all my seeds like cracked open, but then they didn't go anywhere. And they literally drown. Um, but step three or 4, depending on the process, that's the one that's the most important, and that's the one with gibberellic acid, and that's because of the enzymatic process and the hormonal and gene complexes that gibberellic acid is pretty um, it's pretty unstable over a long period of time, and it starts to break down. And so degraded gibberellic acid leads to a gene which is called CSGA3OX, and that gene is not able to fully express itself. And that gene is responsible for synthesizing the hormones needed to signal the next gene, which is CS-DELA, DELLA. And though that gene develops proteins that essentially drop the brakes on growth, right? And so you've got to get the CS-DELA gene activated so the proteins get made so the seeds can grow.
So there's a half-life of it, and the consequences of going too long are that you're in like an even worse case scenario.
If you go past like 24, 36 hours on the soak, you're actually you'd almost be better off to just soak them in some fulvic humic acid and toss them in soil, or just soak your soil with some humic fulvic acid and toss the seeds in there. You'd have better luck than trying to do a 36, 48 hour soak with seeds.
I usually do like full power green aminos and what's the fish brew?
Yep.
Oh yeah. And I just want to make sure everybody in the comments understands that we're talking about germinating old seeds. Like I would say what I consider old is 20 plus years, They've been sitting around for that long. So a lot of what we're talking about is getting around the issues of what old seeds, we're not necessarily discussing how they were stored, but how old seeds just sitting around, how difficult they can be, but there's still solutions to getting them cracked. These solutions might not always be in best practice with certain methods like KNF or, these real strict organic standards. So there are occasions where things like hydrogen peroxide or, using tissue culture are absolutely necessary if you're looking to revive a specific
Someone's
type of,
taking notes.
Yeah. So what I do with a fresh batch of seeds does not necessarily correlate with everything that's being discussed here. But when I'm starting to open up a pack that I've had for 25, 30 years, it's definitely, or that I know is 25 or 30 years old, the process is much more delicate and involved, and I understand that I have to do some things, and then to get back into my regenerative organic type practices, I have to ease into that as well. Because, and this is something I understand from gibberellic acid, talking about the half-life and stuff, but the time and place that you applicate gibberellic acid, and I know I'm screwing that up, but it changes the way
give us the proper pronunciation slow one time. Is it gibberellic?
Yep. Gibberellic.
Okay.
Gibberellic. So if you're applying gibberellic acid to a plant at a different stage in its life, has a different effect on the plant. So it definitely manipulates certain hormone movement, but where and when you apply it in the plant's life has an absolute effect. So you have to be very careful with some of this stuff. It becomes a much more scientific process, especially if we're talking about pulling out the embryonic material and putting that in tissue culture and starting a seed from tissue culture. And if you screw up tissue culture, then your plant is from what I understand, can ultimately forever be in veg and never actually flower. So there's a lot of things when you're talking about bringing back relic type genetics that are ancient at this point. For you really do have to be precise on everything you're doing. So just so everybody kind of in the comments is understanding, this is a discussion on old seeds. Even though we started off with Afghan traits, this is shifted into old seeds.
Now I'm looking up. Go ahead.
Well, I was just gonna say it's a common topic where, I know I've got seeds in my collection that, were either passed down to me from friends or family, seeds that may have been, bag seeds from, Trinidad or Colombia or whatever, that were, from the 70s or 80s or 90s. And then I've got seed that, came from Amsterdam. Like, I've got a bunch of old Skunk One seeds that, were bought in the mid 90s. Some of the first seeds I ever bought. I've got an old strain from Paradise Seeds that I've got a few seeds left of, called Sugar Babe, which was a kind of a spinoff of Cincy Star. Those seeds are ones that are 30 plus years old now, that need, you might get 10% germination if you're lucky. And when you've got 10 seeds of something, or 6 seeds or 3 seeds, like, success is,
The stakes
you've
are
got to do
high.
everything. The stakes are high, right? And some of those, like, um, I'll give you an example. My best friend Chris has the only known remaining pack of OG chocolate Thai that came from DNA that created the Chocolope. Like, those seeds, when they get germinated, are going to have to go through embryonic rescue, right? Like, those seeds are insanely valuable to the cannabis community, because they're the only embryos left, or germplasm left, of the parents of Chocolope. And so, and the original F1s of the OG chocolate Thai, that was used to create, like, those same ones were the ones that Draw used. That if you buy, like, the chocolate Thai from Hazeman seeds, those came from, like, F3 versions. Like, the ones I'm talking about are like the original F1. And so there's certain strains, like Mass Super Skunk. There's still a few, seeds of Mass Super Skunk floating around from back in the early 2000s, late 90s, that are incredibly valuable. So there's some seeds that are so valuable to the gene pool and just to the industry and community as a whole, that it warrants the effort that we put in to try to get these uh, to the vegetative state. And it's a lot of work. One thing I would mention is, after the seed soak, the softening kind of disinfection pre-soak, when you move the plants over to the gibberellic acid, you want to dunk, or the seeds, you want to dunk those seeds into distilled water to briefly wash them, just to kind of get off any remaining minerals or any of the H2O2 that might, exist. And that wash is, just a minute. Basically, you drop the seeds into distilled water and you just swirl them around, and then you strain them out and you get them out of there. When you move the plants over to what I call the hormonal Hail Mary, your gibberellic acid mix. You don't want to, like, have a cup full of gibberellic acid solution. You want to prepare a like a small dish. Really, is I use a petri dish, and I drop those down into that petri dish, and the seeds are not really fully covered. They're allowed to kind of be exposed. The part of the seed is allowed to be exposed. Usually what you'll notice is that, due to the surface tension of the liquid, it'll kind of pull itself up and make a bubble almost
Mm.
over the top of the seed, just due to surface tension. But that gibberellic acid soak is 4 to 6 hours max. Like, I don't care how old the seed is, how desperate, like, I never go over 6 hours, because
And this is from what you
overexposure.
were just talking about, right?
Yeah, because if you overexpose the seed to gibberellic acid or GA3, it can cause the emerging taproot to stretch out so fast that
Someone
the
nailed it.
cells get weak, and they're not capable of sustaining the nutrient transport. Have you ever germinated seeds and then saw that the stem collapses on the seeds and falls over?
Yeah, like
Like,
completely breaks off.
Yeah, like you got a good
Yeah.
germination, and then you come in the next day thinking you're going to see a bunch of healthy seedlings, and all of a sudden, all the seedlings are laid over on their side, and you're like, "What happened?" And you look down close to the soil level, and the stem is just collapsed completely,
It's like
and the
it's either thin or mushy.
yep, yeah.
Yeah.
Now that's usually caused by a pathogen, by either Fusarium or Pythium. Pythium usually, but uh, it can be caused by um, other pathogens or too much water, too much heat. And what happens when too much water, too much heat, the cells try to go too far, too fast. They get weak and they just collapse. That's the same thing that can happen with gibberellic acid. Um, and so 6 hours is the absolute maximum. I usually cut mine off at the 4 hour mark. Some seeds, if they're not super old, will get 2 hours because gibberellic acid it's really powerful, and it doesn't take a lot. And then somebody asked about my uh, mixture and the ppm. I do about a 250 ppm parts per million solution. So what I do and how I mix up the gibberellic acid, you can get gibberellic acid from reputable sources off of Amazon. There's tons of places to get it. It a little baggy. It's a white powder. Um, Wear gloves when you handle it. It's not good to get on your skin. You don't want to breathe it in. It is a hormone that will have negative impact on human growth. It can be a carcinogen
Don't
in
adjust
doses
your
that are
balls after handling.
Yeah.
right, right. So, but just wear some nitrile gloves. And what I'll do is I'll dissolve about 0.25 grams into a liter of water. And before like it dissolves pretty readily in water, but if you want to make it easier for it to dissolve, add a couple of drops of rubbing alcohol to your weigh out your 2.5 grams, then do 2, 3, 4 drops of rubbing alcohol and make a little paste, and then put that paste into the distilled water, and it'll dissolve readily. It forms
And then the
a colloidal
alcohol will
emulsion.
boil off.
Yeah, the alcohol. I mean, you're using such a diluted amount in a liter of water that it's it's a part per trillion mix of
Yeah.
alcohol. Um, but usually and the alcohol will dissolve in the water and then break down because, especially in distilled water. But the GA3 solution is the kind of the magic bullet for germinating old seed, and especially, what the biggest thing is outside of age is how you store your seed. I see a lot of people that they'll hit me up for either just tips on how to rescue seeds, or sometimes I've done the process for them. Um, don't put your seeds in the freezer and then like take them out and thaw them and then put them back in and then take them out and put them back in. Like if you put your seeds in the freezer, which is a great place to store them, leave them in there until you're ready to germinate them, and try to minimize the number of times you take them in and out. Um, if you do have to take seeds out of the freezer, I just recommend leave them out. Like if you freeze the seeds, just leave them out. If you take them out and they get room temperature, don't put them back in the freezer. You risk doing more damage. You can put them in the refrigerator and keep them cold, which will not be quite as good as the freezer, but it works well. Um, and try to keep them like in the refrigerator in an airtight container, and the same in the freezer, because freezers and refrigerators are desiccants, and so they can dry out the seeds unnecessarily. Even seeds still have to retain some amount of moisture, um, and if you could put like I've found some little in the outdoor section at like Dick's Sporting Goods or Walmart. Usually they'll have these like plastic containers that you can get to toss in your kayak or canoe or whatever, that are watertight, and you can usually fit about 50, 60 seed packs in there, depending on what the packs look like. And they're not very large, but they're big enough to store a good amount of seeds in, and they're great
Dry
for storage.
boxes.
Yeah, little dry boxes, they're perfect. But storage is I've seen seeds that are 5 years old that have worse germination rates than 30-year-old seeds because of poor storage.
Yeah, that's a big thing.
Can you
Like
use
white,
willow for all those gibberellic acid scenarios?
um, like a they talking about like a black willow, like the willow tree.
Yeah.
Um, yeah, because so that's an interesting question. Um, there there is a lot of science behind this. So willow are in the Salicaceae family. Black willow specifically, which is used for what they're talking about, is Salix nigra. Um, it contains the precursor to aspirin um, and salicylic acid. Salicylic acid can trigger the same hormone, downstream hormone effects as like gibberellic acid. Um,
Okay, I was wondering about that in that little seed start thing. I was like, is the willow and the aloe willow rooting mixture actually providing a gibberellic acid?
It
yeah,
seems like
it's
they have slightly different
Yeah.
purposes.
yeah, they do. But so it's or if you want to do this, so let's say you want to do because you talked about regenerative farming earlier,
Yeah.
and if you want to stick on that regenerative farming side without having to use lab grade gibberellic acid, which to be honest is often extracted from plant sources, and a lot of times is extracted from willow. Um,
Mhm.
it's a little bit different process than gibberellic acid, but it is a common method used in the horticulture industry by people who really like to stay as organic as they possibly can through the whole process. Um, you know, willow extract biologically helps old seeds in many different ways. The salicylic acid, it's a plant defense hormone that um, that triggers defense mechanisms that lead to a cascade of hormonal complex reactions that you basically you're tricking the seedling into believing it has more gibberellic acid than it does. Essentially, is what's taking place. Um, there's also IBA, indole 3 butyric acid. That's also in willow, and that's a natural auxin. That's a lot of the reason that willow works so well for rooting. Um, but it kind of has that natural anti-pathogen that works very well for old seeds too. And IBA can trigger rapid cell division, similar to like GA3.
Hmm.
Um, and so that really helps the degraded embryos get a vigorous root system going.
So not the
Um,
same compound, but very, very similar behavior in the plant.
yeah, now there is some GA3 in there, but there's also the IBA and the salicylic acid. And so instead of just it's much smaller amount of GA3, but you're supplementing with the IBA and the salicylic acid. And so instead of just using GA3, you're kind of using GA3 plus the IBA plus the salicylic acid, and it essentially accomplishes the same thing.
That's awesome.
Yeah, and I've
That's,
done that extraction. I've went out and
Oh,
harvested. You know,
sorry,
you
Brett.
you go out and you harvest. You want to harvest like fresh, young, green or yellow willow twigs, first year growth. They need to be thinner than a pencil. Don't get thick brown woody branches, but they have much lower hormone concentration. Imagine you're going out to harvest fresh shoots to make like a FPJ, right? Like you
that's
want to
what
get
I
the
was gonna
freshest
say.
shoot. Yeah.
And I wish I literally, like when I walked to the grocery store for breakfast this morning, I walked past three and I was like, God, I should knock on their door and see if they'd mind if I pruned all their little willow branches. Because you can see them. They typically are like really close to like
I'll
the
maintain
ground.
your willow tree.
Yeah, for sure.
Right.
And you can see them and they look really unsightly when you haven't pruned off all those sucker shoots. And every
Yeah.
time I'm just like, God, I could make so much of that mixture, but I think it's kind of like GA3 where the half-life does not last very long. Like you make a gallon, you better soak your whole medium with it because tomorrow I don't think it stores as well. Like you put it in the
Yeah.
fridge and it holds a little bit longer, but it starts to degrade pretty quick.
Yeah. It's pretty easy to make though. Once you get those shoots, you get all the leaves off. I usually will do like a decontamination soak in the H2O2, chop them all up, all those little twigs into 1 inch pieces. And then the best way is to use a cold steep. So place a cup of chopped twigs into a mason jar, and then you cover it with, I usually will do about a cup and a half or 2 cups of warm distilled water. Don't do boiling water or super hot water. And then just let it steep in a dark place for, a day, 2 days. And that water is you'll notice it start to turn like it'll almost kind of be like a light pea color, like it'll get kind of golden, and it'll smell like the twigs. Like you'll have this like earthy kind of smell. Then you just strain out the twigs with a colander or some cheesecloth. Um, it works good. There's different ways to concentrate that if you're using it for seed soak. You can
Yeah.
soak a lot. You can soak a lot more twigs with less water. Like, or maybe and cut the. Sometimes I've even used a mortar and pestle to crush up the twigs, like to macerate them a little bit to help
Surface
improve.
space.
Yeah, to improve surface area, and also it lyses the cells. It breaks the cells open, and that works pretty well.
So I know not necessarily. I don't know if I do this for old seeds or anything, but
Copa, is that your backyard, by the way?
No, that's my buddy's. That was yesterday.
I love it. But my
That's
buddy
convenient.
Miles over with fermented plant extracts, he makes a really killer willow extract, and he basically does like, I don't know if I'd call it a cold ferment because he does it out here in like the desert, so it's a little bit warmer, but he basically, does that aloes soak and then brown sugar and some labs, and it creates an amazing extract that works as a foliar application. So you can literally, like, you spray it on the living plants and it really does help. Like, I don't know how to explain it other than like the plant just seems to get more stacked up. Like its roots seem to get thicker, its branches seem to get thicker, and it's not like your typical silica application. But yeah, I've noticed there's something with willow, even on like a foliar application to existing plants, has like massive benefits. And I wonder if it is like the gibberellic acid replication type thing. But I don't know if using labs as like using that in your solution to try and get like old seeds to pop up. I feel like that would be kind of counterintuitive, but I'm not 100% sure. I've never had luck using labs with starting seeds.
Well, labs, lactobacillus is mostly sorry, I'm actually in the middle of changing the brakes on my truck. So, labs.
Earlier, I had my hands in the back of the toilet because it's it like the water won't stop
literally
going.
Just now putting I just put new back
Well,
on.
sorry. Sorry, I was trying to. I was trying to make him big when he was doing the wheels.
But the going with back to the labs though, the labs is primarily anaerobic bacteria, and so it a lot of the benefits that you get from using labs is going to be in the soil or in anaerobic conditions as opposed to like aerobic conditions, which is what you primarily want for your seeds. Now that being said, I've actually done some experiment where I was using lab and biochar and a bunch of other stuff, and I did notice. So I would encourage people, if you're mixing your own germination media. So like, say you're mixing a little bit of cocoa core, a little bit of perlite, a little bit of whatever it is you mix in. Add about 5 to 10%. Don't go any more than 10%, like 5 to 10% biochar. Prior to adding your biochar to the germination media, put a microbial inoculant and nutrient mix and soak it for 24 to 48 hours. What I usually
Charge or
will add
biochar?
is I'll use a mycorrhizae. It doesn't really matter which one. Pick your favorite one. They're all about the same. Extreme microbes got a good one. Recharge, I think, was from Greenhouse was pretty decent.
Bioag
There's a
has
bunch out there.
VAM. Green
Bioag
Grow
has,
has.
yeah,
Root
I forgot
wise
what theirs
is
is.
killer on
Yep.
Yeah.
root wise.
Root wise.
and so soak that at. It doesn't have to be overly concentrated, but I usually will do about 1.5 times the recommended amount based on whatever the manufacturer suggests per gallon. So if it's a tablespoon, I'll do a tablespoon and a half per gallon. And then I will add the fulvic and humic acid. I will add the labs, and then I usually will use FPJ as kind of my nutrient source. Fish emulsion, also fish emulsion and kelp. Like if I had to give somebody a recipe for a great germination media mix, I would say do a combination of perlite, cocoa core, peat, biochar, and then soak the biochar in the mycorrhizae, FPJ, fish emulsion, kelp, and labs. And I guarantee you'll have some of the strongest looking seedlings you've ever had. Because the biochar holds on to the nutrients. It's a high affinity for the nutrients that keeps the plant from uptaking them too quickly. It's like a slow release fertilizer with the
So
biochar.
let me ask you this. It was actually a discussion I had with a buddy a little while ago around biochar, and I never really went further than the discussion. But there's this, when you use biochar, say you do something screwy in your soil, or your soil acquires some pathogen, is it possible that the biochar is going to retain that longer? Meaning, if we inoculate, if we do the biocharge, is what I call it, where you're pre-soaking your biochar and mycelium and nutrients, or mycorrhiza type bacteria nutrients, if something shifts, is there a possibility that now you have this basically a house for all this bad shit to live in and you almost can't get rid of it? Or is that not a thing with biochar?
Not seen that happen. And reason being is usually the beneficial microbes that are in the soil. I'm not saying it can't happen. I've just never seen it happen. Biochar for a long time. And I usually am inoculating with microbes enough. I'll tell you, there are a couple of microbes that just they grow so rapidly that they outcompete almost any other potential pathogen, and that's Bacillus subtilis. Impello Biosciences has Tribus. That's one of my favorite go-tos for Bacillus subtilis. They've got a couple of great different strains of Bacillus subtilis that are just, they just go to town. And if you give them that carbon source and sink plus the surface area of the biochar will live on, that they will outcompete any pathogen. And they also, as a byproduct of their metabolism, produce basically antibiotics that cause other pathogens to die. And I'll tell you, Bacillus subtilis is a fantastic.
That's a savage skill set.
It's I look at it as microbial warfare. I despise pathogens, and since I've been living in the Northeast, I've had to deal with septoria, and septoria
Yeah.
it is like
It's not fun.
no, it's
So
not. It is probably it's probably single like the worst thing you can have in your garden. Like
oh yeah.
absolutely the worst thing.
I had it
Um,
years ago, and I was growing plants in someone else's backyard, another preschool dad, and they all got septoria.
That's what I love
it,
about where I'm at. Like all those wet bacterias and funguses and diseases that show up are pretty rare out here in the desert. We have like curly beet top virus and a couple other viruses, but we don't have a lot of pathogens out here because it's so dry and so hot. Nothing likes to live here except for a bunch of weird people. But I was gonna ask, have you ever tried growing in just like straight biochar? Like some people do it with perlite and they'll do like a hydroponic type system, clay ball type thing. I've often wondered if
yeah,
you could like do straight biochar and what the results would be like.
I did. We tried this a couple. It was 2018 back in Oregon. I was working with some guys doing some research up there at Rogue Biochar. Um, Grant Sheave, fabulous guy. If you ever get a chance to talk with him, I don't think he's at Rogue anymore, but he very knowledgeable about biochar. We kind of had the same question. One of the problems with biochar is the pH. Biochar, depending on the feedstock, it's not. Oh, sorry, I had a call there.
What
It's not.
What was his last name?
Grant Sheave.
Like S
I
H.
think it's Sheve. Probably one of the most knowledgeable humans about biochar that I know. Like he's a wealth of knowledge when it comes to biochar.
So it's naturally more acidic? Is that what
No,
you're
it's
fixing
very
to say?
basic. It's
Oh,
very
really?
basic. Yeah, depending on the feedstock. Most of the good feedstock is hardwood. And it natural pH, like we did a couple slurry tests off of it. And we've seen pH size like 12, 13.
What? That's nuts.
Yeah. But
Yeah.
what's weird is though, when you mix biochar in a soil that's got like a pH of like say 6.5, and you mix in 10% volume biochar, it has no effect on the pH, which makes no sense, right? Like, because you would think that taking something with a pH of 12, 13, mixing it with a slightly acidic soil, it should, by all accounts, I mean, we're
Buffer it
mixing
up.
10% volume, it should drop that pH down below 7, but it doesn't. So what I started doing is I would pH my biochar, like when we did the just pure biochar grow. I would pH the biochar and try to get it as close to 7 as I could. And I know this is going to sound crazy, but I used Coca-Cola. I found that worked because it's got the
Fucking
phosphoric
get
acid
out of
in it.
here.
I'm not even kidding because it's got the phosphoric acid in it. The phosphoric acid ends up breaking down into an available phosphate for the plant. The hydrochloric, the acidic part of the phosphoric acid, it's cleaved off, and you have an available phosphate. And we did grow plants in straight biochar.
With Coca-Cola.
With now, when I say the Coca-Cola, I wasn't feeding the plants Coca-Cola. I just
Right.
used the Coca-Cola as a initial pH buffer to get the pH down into the right range, because it has sucrose, fructose, it's got sugar in there. That is readily consumed by all of the microbes,
Yeah.
and so it worked. Would I do that again? Probably not, but it worked at the time.
All
yeah,
my drip tubs got clogged.
you know, you can do it. I wouldn't recommend it. It just is it's challenging. I think a mix of, like I see what are they called? The Beto buckets or whatever, where people grow in straight perlite. I see that. You see that a lot in like cucumber greenhouses and stuff. The what's his name? The guy that does a lot of industrial hemp stuff out in Colorado, Colin Clark.
Colin Clark. Yeah.
Yeah,
Oh,
he,
yeah,
he used he used to grow a lot, and I think he had some experience in the past with using those buckets for cucumbers. And I think if you mixed biochar in with the perlite, it would work very well. I don't know anybody that's done that, and I haven't done that. That would be something cool to do.
Yeah. What ratio would you start at? Just a 1-to-1?
I'd probably go a little less than 1-to-1. I'd probably go more perlite than char. I'd probably go. I might go up to 25% char in that mix.
Okay.
Because there's no other really significant organic matter.
Right.
And so
you'd charge your biochar and your perlite the same?
Yeah.
Just soak it all together.
I'd soak it all with like a pre-mix with full of microbes and nutrients, just to kind of, because there's nothing in perlite and nothing in raw char.
Right.
And I've seen people have horrible experiences with biochar if they don't charge it prior to mixing it in.
Oh yeah, I've seen it, Rick.
It's like RO water, basically.
Yeah, it pulls everything out of the soil to charge itself, and it basically
you keep cutting the video off.
2 years to get it back. But yeah, I want to see what's going on in Cocos Grove.
Yeah, show the Super Kush again.
Yeah.
There we go.
But I appreciate everything you've been chatting on, Josh. I think seed germination is some shit that a lot of people struggle with, and they're afraid to ask because it seems so simple, but there's a lot to it.
Oh, sorry.
Yeah, there's there's a lot of information, especially when it comes to trying to get old seeds to germinate. I think their biochar plays a role in that process as well at some point.
Vermiculite. Sorry.
Yeah.
Vermiculite. That's another good one.
Yeah, perlite drives me crazy.
I know, it's like soil herpes. Once you have it, you can't ever get rid of it. Copa, show me those ladies, bro.
I can't right now. I've been teasing.
It's like he's showing a little leg every once in a while.
Yeah, you gotta pull up the skirt a tiny bit.
Some ankle.
I was hoping to get some. What I'm gonna do is take my phone with me. A lot of my spots don't have any service anywhere close, but I do have a couple that.
Just do like a little narrated recording, and then we can upload it.
Oh, that's not a bad idea.
Play it while we're live.
Yeah, I like being live though, because then there's no bullshit, no excuses.
Well, you can do that. So all you got to do is jump out, do a pre-recorded thing on your phone, send it to Peter, and then jump on live, and you can comment on everything you just show us.
It's actually not a bad idea. I took today. This is the first day I literally took off, and I don't know, since the season started, because we got so much traffic going north on the interstate right now, because everybody's coming up from New York, New Jersey, Connecticut, Rhode Island, Mass, everywhere. So I said, fuck it.
My daughter yesterday went up to camp in Maine, and so I had to drive up. Fortunately, not all the way. I saved myself about 4.5 hours round trip.
Yep.
And my mom was my sidekick in the front seat. And she was complaining about the traffic and the 18-wheelers.
So would you go up 95?
Yeah, just straight up 95.
Yeah, man, I'm 10 minutes from 95. You should have let me know. I could have popped over and got a coffee with
And
you or something.
smoked my mom out.
Yeah, well, I don't smoke during the day. That's the thing. So
You got
you
3
would have
more
smoked.
hours, and then he'll be on a plane.
Yeah, exactly.
Yeah. You could come all the way down to Rhode Island.
Yep. Yeah, Rhode Island's not bad. I've been to the Viking a couple times. Newport.
Yeah, no, it's very sad. The ocean's 70 right now. The waves, I think that it was like 4 to 9 foot swells.
Yeah.
It was awesome.
Yeah. All right, let me ask I forget who it was talking about Septoria.
Oh,
When's
that
the
was
first
Josh.
time you've seen Septoria up in the Northeast? Because I've literally last maybe, I don't know, 2017, 18, 19, I saw it here and there. Before that, I never saw it ever.
So I moved. I'm originally from Alabama. I moved out
Yep.
west, 2014, 15, and then I moved full time here, 2019 in Massachusetts, in West Mass.
So you're saying you brought it with you? I'm just messing with you.
No,
Yeah.
I'd never
The
seen
day
it.
I
I'd
showed
never
up,
seen it.
actually.
God damn it! Now we know.
No, I'd never seen Septoria
Okay.
until I got here. I had
Yep.
We were doing industrial hemp farming for CBD extract with a bunch of farms in the Pioneer Valley, and one of the smaller farmers that we were working with, I say smaller, he had 10 acres, and he had done plastic mulch, and he had a
Yep.
beautiful looking setup, and it was basically in his backyard, and he's up close to South Deerfield, and
Okay.
he one Sunday afternoon he decided to use his riding lawnmower and cut the grass between his rows, and he got to thinking, he's like, shit, I'll just spray that grass mulch and have it as an extra mulch layer, and this is like mid-August, so we're just getting into flower.
Perfect timing.
And and he calls me on, I don't know, like Wednesday after he did that on Sunday, and he says, hey, can you come out here and look at my plants?
Some
It's like
spots.
something's going on. I don't know if it's bugs or what.
Yeah.
And so I get out there and I see these like little spots, and I'm like, I don't know.
Starts low and goes high.
And so that was like Wednesday. I took a couple leaf samples with me. One of the guys we were working with was a professor at UMass Amherst in the Stockbridge School of Ag. So I carried the leaves over to him, and I was like, hey, have you ever seen
What is
this?
it?
Like, I'm kind of stumped. What is it? And he just looked
Septoria.
down and shook his head, and he's like, that's death. That's what that is.
Yeah.
It's response. And I was like, what?
Yeah.
He's like, that's Victoria. He said, he directed me to a page on the Stockbridge School. They had done, years of studies looking at different copper-based fungicides, regalias, Marone Sciences.
Marone.
Regalia,
Yeah, no, I use that.
Yeah.
So
they're great preventatives, but once your plants get Septoria,
there's no one getting it.
The only way is to go through your garden and literally remove every leaf that you have a spot on, and you have to destroy it by fire, and then even then you maybe got a 10% chance of success. I watched this guy's field go from one of the most beautiful 10-acre plots of plants to having about 2 fan leaves in 10 acres over about a 2-week period.
Well, let me ask you a question. How many strains of varieties or F-gens were going in that field? All the same? Was it cuts off the same plant? Because I've had some plants right next to other plants that have zero resistance, and some of them are like Superman. They won't get Septoria. Same for TM. You get a plant beside another plant, one's covered and one has none. So there's got to be some kind of study somebody can do to figure out what the actual resistance is in the plant. I'm not a scientist at all.
Well, then also the soil after, right? So can you remediate it over time? Like I use
Yeah.
those and I hit the soil and I actually eventually reuse that soil and I've never seen it again.
I have a buddy that's into KNF and everything else, and I'll tell you, his hugel beds, you could have Septoria anywhere around, but the plants that are growing on his hugel are impervious. Now you take that same cut of whatever cut he's running and put it different part of his yard, and it's going to be run over with Septoria. So
Yeah.
I don't know. It's got to be.
I'm doing hugel beds here, and I've noticed the same. I had plants 2 years ago that got Septoria. Everything that was in the hugel bed, completely fine, and
Yep.
growing 3 feet away with plants that just I ended up having to cut them down. They got so bad.
So that makes me think that the ones in the hugel bed are getting what they need for preventative medicine, be it microbes, or the good microbes, and
Well, that's
probably
what
has a
makes me wonder. So,
good
like,
immune system.
when you talk about a hugel bed, it kind of goes to the same thing as the biochar, is you house a
Yeah.
shit ton of really good bacteria
Yep.
that ultimately outcompetes all the shitty stuff.
Yes, you do.
I mean, that's pretty. I mean, that was like day 5 when I started getting into KNF. That was what turned me
Yep.
on to it so much, was like, oh wait, you can just you don't have to
Exactly.
you don't have to treat this. You can just outcompete it.
Out hustle it.
More bodies equals winning. Like,
Yeah. Yeah.
So that was like what drew me to it, and then it was like, and that kind of like what got me into wanting to teach people was, there was a point in my cultivation experience where it was, you either do this or you do this. There is no blending these two methodologies, which is what I loved about like Josh coming on talking about old seeds, is there's always room for you to be able to go, oh, you know what, we're going to put a little science tech to this. We're going to take a little bit more control. And then when we have it, we're going to let it go back to nature and apply all this stuff. And yeah, the KNF thing was such a big kind of like relief to know that you don't have to fight off everything. You can just let something else do that for you.
Yep. Yeah. So, I mean, as far as, was Septoria always around?
I mean, I don't know. Like, I don't know if there's. I need to look into the history of it. It's been. I mean, it's been here since I've been.
Is it? I mean, so I know PM is cannabis, or the type of PM that hits cannabis is cannabis specific. It's not the same type of strain of PM that hits, say, squash or pumpkin leaves.
Yeah.
Correct? Am I right? Okay, so Septoria, is it cannabis hemp specific, or is it? I don't know anything about it. That's why I'm asking.
As far as I know, Septoria will impact tomatoes, potatoes, and a handful of other plants that are commercial.
And the same strain of it.
That's what I was asking. Pretty much.
Yeah.
Yep.
Yeah, it's the same. It's the same strain. Unfortunately.
Okay. That's interesting to know. I have tomatoes and peppers, all kinds of tomatoes,
Everybody's
all kinds.
getting it.
But it's so the trick is to Septoria is mulch. It's soil borne. What causes Septoria is either you cut grass and sling the dust from the dirt onto the plant, and which has the spores in it, or if you don't mulch around your plants and it rains and the rain splashes up. A lot of tomato growers get it from not mulching under their plants, and then it rains, splashes up, and then they lose their crop.
Yep.
And so that's why I've started mulching religiously. Like, I mean, I don't grow now unless I'm mulching. Like, it just, that's just part of it. If I'm going to take the time to put plants in the ground now, I'm putting down mulch. Wood chips work pretty well.
That was a tough lesson learned.
Yeah. It was.
So you're saying Septoria is pretty much inherent in the soil everywhere around, and you just stir it up and get it? It's always been there, pretty much?
Yeah. It's from what I understand about it, Septoria has been around for a long time.
Since the dinosaurs?
Dormant for a long time.
Yeah. Well, I'll tell you, I've never seen it in the swamp. I always bring in my mix, though. I bring in Pro Mix, either HPCC or HPCC with the mycorrhizae or mycorrhizae, whatever it is there. And
Yeah.
I don't ever see it out in the swamps at all, and it's always humid out there. You don't get a dry day in the swamps. So I'm not
Yeah,
sure,
it seems to prefer sandy loam soil.
but you don't have that in the swamp for sure.
That's interesting.
You got watery duck shit, maybe.
Most everybody that I've seen that has trouble with it are in the Pioneer Valley where they have the Hadley loam, and a little bit out in the Berkshires, and then over on the Cape. Like I had a buddy that was growing out on the Cape, and
Yeah.
he got septoria really bad, and I was like, "What kind of soil do you have out there?" Just out of curiosity. He's like, "Oh, we got it's really sandy," and he didn't mulch. Just had plants in the ground, and it rained, and 2
Yeah.
days after it rained, he's got spots.
So do you think there's a possibility that it could run not in pure amendments, but in amended soil that they sell, the blends, like say, Stonington blend, or I don't know what the 707 blend, the little camo bags, like, any of those places that process dirt, because they could just be handing out to everybody everywhere. Not even knowing.
True. I think with local brands, I think it's an issue like Coast of Maine.
Yeah, that's exactly what I'm talking about. Stonington blend. Yep.
Well, and that's where I would look into
like,
the compost.
Out on the West
sources.
Coast, I don't think it's as big of an issue on the West Coast. Though symphyllins are an issue on the West Coast, which I would rather have septoria than probably symphyllins. But I think Route 707. I don't.
Yeah.
I'd have a hard time seeing that, but I mean, it could. That's the problem. Like,
those soil
we're not
companies,
staying local.
Well,
if those soil companies are using, like municipal compost.
Yes, that was the next thing I was going to say. Mr. Fox composting. They do a lot with a lot of the people around here.
So much where you're just like, and they like even here we have this shit called Mesa Magic, and it's literally like what everybody in the valley hauls to the compost dump.
Yeah. A year later or 2 years later, yeah.
And they're like it's good to use in 6 months because we compost at high temperatures, and it's like no. And I can't tell you how many soil companies use that as their compost because it's cheap, and then they stack a shit ton of sawdust in their soil as filler. I know 707 is bad as that. Like when I was working at the garden center, dude, I remember having to huck some of those 3 cubic foot bags and restack them, and when I was done, I had splinters in my arm.
That's bullshit, yeah.
And that's from a sealed bag. So I'm like, no, thank you.
Well, let me ask you another question. A lot of the blends, the soil blends, like back in, I don't know, late 80s, early 90s, I don't know if ProMix was around. It might have been, but I used to use Sunshine Mix, and Sunshine was number 2 and number 5 were the ones I'd use. And those were more peat-based. When did cocoa come on the scene?
Oh, God, cocoa. People started using cocoa, like, I think I really saw it blow up in, like, 2012.
Okay, all right. So, I mean, like on the commercial weed scene or, cannabis scene.
Yeah, that's really, like, it didn't take long for the commercial folks, because there was a lot of rock wool and soil stuff, especially here in Colorado. A lot of rock wool and soil stuff going around, and people realized they were having, like, a lot of issues with, like, salt buildup and fertigation issues, so they found that cocoa just drained so much easier, and they could feed more precisely. And
growing in cocoa was like skiing through powder.
Versus
on like ice and rocks and.
Yeah, it was a thing like early, like I would say like 2010. People were fucking with it a little bit, but it didn't really take off until the commercial scene had like really like fertigation specialists running through and going, no, this is going to be the best media for your roots and nutrient transport and all that. And that's when everybody, people like Athena started popping up, where there started to be fertigation companies that like really showed up with shit that was designed for cocoa, because like there was a big hang up in the early days of cocoa because nobody knew how to really buffer it properly. I think there was a few people like Dutch companies that were doing good. Now like Canna, they were like one of the first ones to really like hit the cocoa scene with like a really consistent high quality cocoa product. But again, you had to have their line to work along with their buffer. And when you start deviating too much, then you start ending up with all of the issues that you see with cocoa, where it's like retaining a shit ton of calcium and then all of a sudden just fucking dumping it. Like, so yeah,
Well,
so it's interesting though, because I've seen a big resurgence in like this living cocoa movement where people are starting to take like really good, high quality controlled compost and utilizing it with cocoa instead of peat, and the oxygen amount that gets to the root zone is blowing up.
Well, you
And
can call it loco.
Yeah, I mean, it's nuts.
Living cocoa. Let's call it
like
loco.
Loco. You know, I think you just coined something. You need to start bagging it up, buddy.
There you go. Local soil. Locosoil.com. It's already taken.
Hey, whoever's talking about my post office, my days are off Thursday, Friday. That shit runs all day.
Too funny.
I saw it in the comments. My post office is never open.
Oh, I don't even see
Cheddar
the
Bob's
comment.
talking shit.
Yeah.
He's a fellow postal employee.
Post offices. I don't know who's an employee here of the post office.
Oh, that would be me and
Oh,
Cheddar Bob.
geez, dude. I'll tell you right now, people over here, they need to broom, sweep, clean, everyone. Everybody's so lazy, waiting for retirement. It's a shit show.
Yeah.
Complete shit show,
That's
dude.
anybody that gets into the post office.
Yeah.
We call
Well, you
them
seem pretty
lifers.
normal. You seem pretty normal.
Dude,
People
I've
here are
only
just
been in it for, like,
fucking lazy.
Yeah, I've been in it for 3 years, but I have a different mindset because I'm on like an FMLA restriction, so they can't force me to work more than 40 hours.
Cheddar
a week.
Bob, that's what you need.
Gotcha. Yeah.
And so while I'm there, I work my ass off just to make sure, so not a bunch of other people have to put in a shit ton of overtime. But it is kind of unfortunate that there is a lot of mandated overtime. So I've worked with people who are like, "Yes, my only day off this week." I'm like, "Fuck that." FMLA.
Yeah, that's not what I'm talking about. I'm talking about, like, I'll go in and there's the guy's a postmaster. I should say the postmaster's a male, but there's two women that work there. There's an older woman. I'll come in and I'll be like, "Hey, how you doing?" Say her name, and she'll be like, "Oh, we got 140 Amazon packages today." Yeah, if I like, okay, that's your job, like,
Right.
carry on.
Somebody else will finish it when you're not there.
Yeah, no, they tried to close the post office down early for lunch one day to sort Amazon packages, and I drove about 2 hours off of one of my spots on purpose to grab the shit I needed, and they were closed. And I was like, "No, that's not how this works." So, yeah,
I have the opposite. The women at my post office love me, and I see them 6 days a week.
Yeah.
No, I see mine 6 days a week, 7 days a week, and I wouldn't say they probably love me and hate me.
Well, I'll tell you, somebody on the other side of the counter, like, I love friendly regulars. Like, even if there's a line, like, we can shoot a quick bullshit session in 30 seconds and have a blast. And usually I can rouse up all the other clerks, and then the next person in line is laughing. Like, I just have fun with the behind the counter shit. Throwing packages is kind of a pain in the ass if nobody else is putting in the work. But, dude, on Sunday I go in, I'm pretty much by myself, and I throw like 5,000 packages, 6,000 packages in a day.
That's a lot.
In an 8-hour period. So it's
so I hate to say it, but where I am, it's tiny by comparison, probably. But if they get a bag of Amazon packages, it's half the size of a pallet that they
this guy right here may be your mail carrier.
Smoke shots coming out of your ears.
I recognize that voice.
Yeah.
It's like the shoe shine guy in Caddyshack.
Oh, geez. Don't talk about Caddyshack. I hate groundhogs. I'm dealing with a couple of them right now.
Me and Cheddar Bob are going to make a strain called the Boys in Postal Blue.
Oh, jeez.
So after the calendar.
Yeah.
The Boys in Postal Blue calendar.
Yeah.
So I got another off the subject question about what's happening in November. It's probably been talked about a whole bunch, but I haven't been around to take part.
We've been trying to expand. Actually, we did a little session on it, but I'm hoping to get Omar back on, and then who is the economist, Peter?
Bo Whitney.
Oh, yeah.
So
reach back
I guess I guess my question is, how many Mark Emerys do you think are going to be created once that passes?
Oh shit, all of them.
You think? But, I mean, don't you think they're going to go after like Brothers Grimm or someone huge and not the little guy, or do you think they're going to go after everybody? How do you think it's going to happen?
You know, I think they're going to be looking at anybody who's not willing. Like when it comes to companies, I think it's going to be whoever's like really trying to hold out and make a stand.
Like I'm selling high-end bird seed. Come and get it.
Yeah. And,
okay.
And
it's the dumbest thing.
So
stupid.
It is.
Yeah. Well, you know.
Brothers Grimm is a good example because right now he's hitting it hard in the mainstream scene.
Yeah,
so
and I know they're huge. That's the only reason I mentioned them.
Yeah, and he's been doing his thing since like the late '80s, early '90s, I think, back in Dutch. So he's kind of been established.
Yeah.
And
is he even doing anything? I heard his wife was doing most of all the shit now.
You know,
no, I think he sold it.
No,
right?
no. Him. So. If I met him like a year or so ago, and I can't speak on this because I haven't really talked to them, but just from what I've seen, he's got some neurologic stuff going on. Homeboy has like early on, he's got like Parkinson's or some shit, dude. He was shaking real bad. Yeah. So I think his wife is running things, like doing all of the work. And I think he's literally just guiding, like, this is what I like. He'll go in and smell and select, and she does all the work.
Film this here, please.
Yeah, and then she's the thing that I've noticed is she's kicked up their marketing. Like they've done a bunch of collabs with the Trailer Park Boys. They're tight with Montel and doing like a collab with them. So he's in this big which fits because his whole brand has been like designer jeans kind of thing. So. It fits. Now, if he doesn't like link up and do the whole DEA whatever that they're expecting everybody that wants to be legit happens, I guarantee you they're going to come down on him. They're going to look at him as somebody to make an example of, right? Those are going to be the Mark Emery. So anybody who's like, oh no, I'm too scared of this, I'm shutting down. Anybody who's standing up and they're like, no, fuck you, free world. Those are going to be the people that really, I think, get an example made of versus like dudes like me and Peter who are just kind of
I hope so, Rusty.
Well, I'm just going to say, do you think they're going to come after the little guy breeders like us, or do you think they're going to come after the seed banks, like Peter? Or do you think they're going to come after like,
that's what I think Microbial Solutions will slide under the radar in the sense that we're not, dude, we're a splash. Like, that whole company is just a drop in the bucket.
Your Honor, I'd like to present exhibit number 3.
Right.
This YouTube conversation.
These idiots talk exactly. They fucking knew.
But I mean, going off of like a conversation we've had with Omar, like the states still have their
right to flex their.
Yeah.
So postal employees at the front line have the right to ignore laws.
Whatever we're doing, whatever I'm doing in Colorado, whatever I'm doing here in Colorado, if I am working within the Colorado framework, then technically the feds can't do anything, just like they couldn't do anything since this all started, really. So it all depends on like what level are you trying to play. I think Brothers Graham, Ethos Genetics, like they're pushing for this national brand shit. So if they're still continuing to push for the national brand shit, then they're gonna have to fall in line because they're too big.
Fuck around and find out.
Yeah. And
so I guess
they're just like fucking whatever. Get out of here. Nobody cares. I'll make $20 a week.
I want to throw a couple of questions out there. So let's just say everything passes and it's illegal, right? What if I went and got my caregiver's license and sold seeds to people that had their medical license, or not medical license, medical cards? Would that be okay? Or could I still get in trouble sending them through the mail?
To Omar, and that's what I thought was interesting. I'm like, so if nobody, because the thing is like some of these companies have to register with the FDA or DEA in order to become like medical cannabis.
Well, I think as soon as it hits the post office, it's federal, right?
That's what I was going to say. So there's, but then you got FedEx and UPS
through UPS and I think
but FedEx and UPS. The other thing is though, they don't need a. Yep, sorry.
Well, just like the hemp paperwork, when the hemp bill went, like you could send any flower as long as there was some kind of fucking paper that said this tests under 0, right? So the same thing, anything you send is going to have to come with like paperwork that basically says we're partnered or signed up or registered through the FDA, so this is considered medical cannabis. Now, once the patient, right, like me as a medical patient with a card, every weed that ends up in my possession is now medical cannabis. Whether it comes with that paperwork or not. The way it's kind of way I understand it to be written is everything in my possession is now medical. Does that mean everything I produce is medical? Does that mean because I'm a medical patient, if I produce it, if I fly around the country with it, it's considered medical, right? Yep. So
that goes.
If you land in a state that doesn't have medical laws, now you're up to their discretion. The Fed shit doesn't protect you until you. That's the way I understand it.
So it's almost like carrying a firearm. If you have a license to carry in your own state, it's one thing. If you go to a state that has whatever open carry, constitutional carry, whatever, you're subjected to their rules. All right. So I guess if you're mailing something through the USPS, it's federally regulated. They need a warrant to get in a package. If you send something through a private carrier, FedEx or UPS, technically they don't need a warrant if the carrier is willing to just hand over the package.
Exactly. Yep.
So you know, 6 in one, half dozen in the other, right?
Yep.
That's interesting. Yeah, I was like, damn, like which one's?
No, I mean, hey,
which one's a lower risk?
What do you like, blackjack or fucking roulette?
You know what I mean? And I have to tread very lightly on this because, God, okay,
you're on camera.
We're gonna use the ladies at your post office for example. They have to close down early.
We need what was the original postal service with horses, right?
Yeah.
Pony
Express.
No day nor night, nor rain nor snow.
I'm just gonna put this there. Those ladies at your little ass post office. There's a process where they have to scan each package. Do you think they are? Beep, beep. Nope. Nobody smells every package coming through. There's no X-ray that the packages run through. Postal inspectors will open your package with a warrant if there is cause. That's
if it smells like RKS.
Right.
Yeah.
So you gotta stay away from like
unicorn.
I'll tell you in today's climate, if that package is detected, 80% of the time the postal inspector will not know about it, and the detector is at home having a relaxing evening.
There you go. Yeah, smoking some good herb too.
It ain't getting reported. You know what I mean?
Yep.
But it's a really interesting thing in that sense, like the amount of work that goes into it. There was one, this is kind of wild, not cannabis related, but a truck driver who drops off mail at one of the locations I work, he showed me a picture of a package that went through the mail stream from Denver, or it was going from Colorado Springs to California, and there was a sticker on it that said suspected fentanyl, and it hadn't been opened yet. So I don't know where they found that in the mail stream, but occasionally.
But only put a sticker on it and let it keep going for the next person to deal with.
Well, you could just drop them off with those stickers just to scare people.
Oh my God! So once it was identified, like I don't know, I think the postal inspector put it on there because once it's identified, then it gets basically intercepted. The way I understand it is they'll actually hand it over to the police and then work with them on a controlled delivery. They'll surround the house, watch it, let the package be delivered, and then whoever grabs the package is getting arrested.
Yeah. By the way, Jeff, you need, maybe we can do a day-long session on the inner workings of the back office of the postal, like where I don't see it, because I'm always like, when I send stuff, I'm like, who's checking for what? Like my little tiny packages when it goes through customs, like.
Yeah, it's chaotic, bro. That's all I
All
can
right.
tell you. Is
Well,
it's
that's
so
my hope, is
chaotic.
that they're just so overwhelmed that they just don't really give a shit about anything, especially
So-
if it's small
That's
and thin.
exactly it. That's
Another-
exactly it.
Quick question: as far as the seeds are going to be illegal, or the sale of seeds are going to be illegal, or both?
The transport of seeds across state line will be illegal.
Okay, so the seeds themselves won't be.
As long as your state has recreational
Will not be
cannabis
legal.
laws.
So I could potentially or theoretically start a seed bank in Maine and sell to my
Oh.
customers in Maine, and nobody gets in trouble.
Technically, yeah.
As long
But
as
you
I don't
could mail
use
it
the
through the
USPS.
USPS.
Exactly. So if I use a
Right.
private carrier and did it to me, now what about? Well, I was thinking maybe donating seeds. So donating seeds across state lines is the same as selling seeds across state lines, correct?
Any travel, and I'm not a
Got
lawyer,
it.
but any travel.
Okay, no, I hear
across
you.
state line. Yeah,
I'm not asking for legal advice.
Anybody listen. It's more anybody listening going,
Yeah.
you know what he said, but
No,
the way
we're not
I
lawyers.
understand it, yeah. Any
But-
travel, any movement
Yeah.
across state line, it's no different than if I have a bag full of seeds. Are now think of seeds like a bag full of stank ass, right?
Yeah.
Like you just got a bag full of dank in your pocket. That's what seeds are now. So in Colorado, because of where we're at in our recreational, our medical climate, the only way I can now legally sell seeds is as a medical cannabis caregiver and
Yeah.
sell them to medical cannabis patients.
But you can
Now-
do that. That's
But
okay.
now- yeah.
Yep,
But that's
good.
the only way I can sell them.
So let me ask you a
So-
question. And with that being said, if a dispensary which has a medical license carries, my seeds or another breeder seeds, and then they sell it, can they sell it to across state lines to a medical customer? Or is that no go?
That's going to determine in your state, and I've been sandbagging on that for a little bit because
Okay.
that's kind of my goal. Is I'm hoping to
Yeah, I just
do
figured
the license. Yeah, no, I'm hoping to do the licensed caregiver thing.
Yep.
And then produce seed and sell them to the dispensaries that have medical patients.
It's going to be interesting to see how this all shakes out. I'm sure there's people running around behind the scenes right now to try and figure it out and capitalize on it.
Oh, it's nuts. And that's why I've been
You
trying
know?
to get lawyers and economists in on this to try and figure out like what the climate is in general, because this is all coming out and relatively quickly.
Oh, it's
happening.
It's going to be happening sooner than we understand it's happening.
Yeah.
You know?
And so trying to get, a firm plan on a foundation that's like, okay, I know how I'm going to navigate this. It's difficult when like how long? It was like a month ago we interviewed Omar and he was like, I'm just waiting. This is somebody who studies the law. He's like, I'm just waiting. We have to wait till all of this language is written, and how he was under the impression that the medical shit, the Schedule 3 was not affected by the hemp laws, but the way the hemp laws are written, they're kind of like mashed into the Schedule 3, but there was no definition. During our interview, there had been no definition on any of this shit, so it's all left up to speculation. And it's
interpretation. Yep.
Yeah. So it's kind of fucked right now. Like we're all still trying to exist and move, but we're all doing it so delicately that you can't continue to make money because marketing puts you out there, right? Like that goes back into like the Brother Grimm shit where it's like, oh, these motherfuckers are big. They're on the radar now.
So the other question or angle would be, partner up with seed banks in Europe, make one shipment out of the country, take your chances, and have them sell it.
And that's been an option I'm exploring too with Brother Mendel as he retires. I've been kind of, in the background chatting with him, like, what other countries might be viable markets. And I think you're going to see a lot of that, actually. You're going to see a lot of American companies dipping underground. Everybody's going to think they went out of business. And then you're going to run over to like South Africa somewhere, and there's going to be a shit ton of this dude's genetics somewhere. Like that's what I think is going to happen.
The microphone, something happened to a microphone or something. I can barely hear anything.
What's that?
Oh, did my
So
mic?
Can you guys hear me? I can't hear you guys. Must be me.
Yeah, no, I can hear you. But
The fuck
why
did I do?
don't let me mute. Yeah, there we go. Why don't we call it there? Because I got to catch my flight
Yeah.
back to the West Coast, so my little fingers can start packing up orders.
I got a guest coming over, and then I got to prepare for a cannabis judge meeting. But this was a fun combo, dude. I had an absolute blast. Thank you, Josh. You provided a lot of
Oh,
really good
fuck. The
shit.
one thing I wanted to go through was the this thing. So
Oh, we did
Josh,
that
next
last week, bro.
time.
Oh yeah.
Oh, we did. Oh, we
Yeah,
already
we did
did
that
it.
last week. Yeah.
Totally forgot about that. It was
We can do
memorable
it
to
again,
me.
though. We can do it again.
All right. Cope, I was just saying to these guys, let's call it there because I don't want to
Okay.
miss my flight out of Providence Airport.
Don't want that to
so,
happen.
No.
No.
All right.
But Copa, it sounds like you've got a lot of good questions. I want to get you on with a lawyer,
we had all
basically.
these.
Like, I want to get Omar in, and you pop in, and I think we can. The closer we get to November, I think he can start to outline what some of this is going to look like.
Yep.
And it's just finding everybody's dates, and then yeah, Bo, I would love to have him on if we can ever. I know he's kind of been a little bit of a flake on Peter, but yeah, dude, you'd be great for that little sesh because I think you've got a lot of solid questions that
Most
either
inquisitive.
Well, hey, the other thing is, I literally don't give a fuck. I'll be making seeds and getting rid of seeds for a long time. So
you know what I mean?
more than half my life already. So it ain't stopping anytime soon. For the record.
That's what I mean. Like, I remember there was Seed Boutique and Seed Bay, and he brought up Emery. Like, we've been doing this shit for a long time. Like,
Yep.
Whatever. Like,
Nope.
they're they're crazy if they think it's gonna work. Like, it's not. There'll be such a backlash from the states. I mean, effectively, if you're a licensed cannabis operation in any of the states, you're stuck with what you have forever.
Yeah.
Some states prohibit breeding. Some states give you a 60-day window to bring in genetics, and after that, you can't do anything. The other states that allow you to bring stuff in, you can't do anything. So it's it's.
So what we should do, what I'm going to do, I'm going to get together some methodology of how to ship seeds the way I used to ship them back when I'd go to Amsterdam or Montreal and get them back in here. The million different methods. Piece of corrugated, those for sale signs, the real estate ones, the plastic ones. Those are perfect.
Great.
Get those little Q-tip, piece of cotton, one seed, piece of cotton, another seed, piece of cotton, another seed. All that good stuff.
And the spine of a CD case.
We used to do hash back from Amsterdam that way all the time. The Temple Ball, Lebanese Blonde, Lebanese Red, right in the spine of the CD. The good one I used to do is I just wear a pullover hoodie sweatshirt, and the tie strings, I'd untie the knot and I'd jam a few seeds up in there and just wear them right through fucking customs. Didn't even give a shit.
All right, yeah, that's going to be our next show for sure, is how seeds were brought over.
That sounds good, right?
That's fucking legit.
I like it.
Yes, sir. Show and tell.
Yeah.
Show everybody what they need to know after November. You didn't see it here.
Nope.
Peter left.
Yeah, he's got to go catch
we're
that
just
flight.
we're just talking about what we used to do, not what we do or what we're going to do, but what we used to do.
Oh, listen, I've been doing this shit since '86.
Dude, I'll tell you what though. There's some stuff you can learn from this Gen Z shit, because I've been watching these fools post stuff on how to legally deal with flock cameras.
Really?
And they're like, they're not telling you how to disable a flock camera. They're just educating you on how flock cameras work and what.
Like shining a infrared light in it.
Yeah, you know what I mean? Well, it's beyond that, dude.
I know.
This morning. Holy shit, this guy was talking about like how those things work, and he was just explaining how like radio disruptors affect them. But he was like, I'm not telling you how to make it. I'm just telling you this is how it works. And I was like, okay, we got to do that for weed now. Fuck.
Yeah, absolutely. That sounds like a good plan. I'm gonna get rolling here too. This is my only day off, so I'm gonna probably try and enjoy some of the cooler weather we got.
Oh yeah, go get it, Copa.
But so hey, good talking to you guys.
Yeah, you too.
All right, brother. Have a good one.
You too.
See you
Bye.
guys.
Later. Are you out of here too, Josh? I think I'm gonna jet as well.
Yeah, I just was finishing up. It's funny, I just changed the rear brakes on my truck during this whole event. So I was multitasking, but yeah, no, and yeah, next time you guys are on, let me know. I'm happy to jump on. And it's
Oh,
been
for
a
sure.
pleasure, and
Yeah,
always enjoy it.
it's always a blast. I love chatting with you, Josh.
Same. Well, you guys, I don't know. I can't really see my screen. The sun's shining right in it, but I don't know who all's
No,
still on. But
we got you. It's just me and Lobster at the moment. I think we're getting ready to
oh,
dip out
cool.
too. Yeah.
Yeah, I know. Yeah, we gotta. I'm trying to convince Peter to have a little gathering at his place down at brother's place there, the family spot, whatever, in Connecticut. That was fun.
You know, I've
Maybe
been
do one
wanting
of
to
those.
take, yeah, I need an excuse to take my wife out that way. She's got a friend moving to Maine, so we might have to do something like that just so I can schedule an out east vacation.
Yeah. What are you in Washington, right?
No, I'm in Colorado. I'm like,
Oh, Colorado. Okay. What
yeah,
part?
right on the Colorado. It's Grand Junction, Colorado. So I'm right on the Colorado
Oh,
Utah
Grand
border.
Junction. Well, yeah. I used to. I spent a lot of time around Durango, Bayfield area.
Oh yeah,
Uh,
yeah,
yeah. So I know Grand Junction well.
yeah. It's a fun little place.
Yeah. There's I used to go Grand Junction down to like Moab, and
Yep.
that a little alien cafe right there off of the interstate
Yeah.
is always to trip me out. It's probably
Yeah.
like a convenience store, but it's got like UFOs and shit painted on it.
Oh yeah, 100%. It looks like something out of a movie. Like you're just driving down the highway and all of a sudden you see that thing off the side of the road. It's kind of a trip.
Yeah, it cracks me up. But well, you guys do well, and let's do it sometime.
Definitely appreciate you, Josh.
See you guys. Bye bye.
All right, thanks. Later. Thanks, Lobster.